Hello guys, this is Dr. Karaki. In this video, we're gonna solve an exercise on protein sequencing, aiming to elucidate the disulfide bridges. We have the following exercise. The treatment of a polypeptide with 2-mercaptoethanol yields two polypeptide chains, X and Y. To remind you a little bit about 2-mercaptoethanol, it's a reducing agent used to reduce the SS bonds located between two cysteine existing in two different chains, and it's called interchain bond, or between two cysteine located within the same chain, and it's called interchain SS bond. In this exercise, treating the polypeptide with 2-mercaptoethanol yields two polypeptide chains. This means that the original polypeptide is made of two subunits. It has a quaternary structure, and that the two subunits are linked by SS bridges. The chain X and Y are made of the following amino acid placed in their correct order given. Since the polypeptide is not subjected to an acid hydrolysis, we have just broken the SS bonds. The next step is to treat the polypeptide with chymotrypsin that yields fragments of amino acids. Of course, the amino acids in the fragments are not in their correct order like in the original peptide, since they are obtained after an acid hydrolysis. Now, using an endopeptidase like chymotrypsin will help us to identify the location of SS bonds between two cysteine molecules. Let's see together how to solve such an exercise. Chymotrypsin cleaves at the carboxyl end of aromatic amino acids. So it cleaves the peptide bond after phenylalanine, tyrosine, and tryptophan at these cleavage sites. Let's verify now the fragments obtained one by one to discover which two cysteine molecules are linked by SS bonds. The first fragment obtained by the action of chymotrypsin is alanine phenylalanine. This is how chymotrypsin cleaves after phenylalanine, and this fragment is released. The next fragment is cysteine glycine lysine. When chymotrypsin cleaves after phenylalanine, this fragment will be released. So far, we haven't encountered two cysteine in the same fragment to think how they are linked. The third fragment is asparagine, 2-cysteine, methionine, and tyrosine. Now you see we have two cysteine, so they must be joined by SS bonds. But let's see if the two cysteine exist in the same chain or in two different chains. The fragment contains asparagine and cysteine, and here they are, these two amino acids. Now we must look for the other amino acids in the fragment, the methionine, tyrosine, and cysteine, and here they are in the same polypeptide chain. So what we understand from having them in the same fragment is that the two cysteines are joined by SS bonds, like this. The next fragment is cysteine, 2-cysteine, leucine, 2-tryptophan, valine. If we look for these amino acids, we find them distributed between the two chains. So they must be joined by the cysteines. Because if they are not joined by cysteine, we should have cysteine, leucine, tryptophan in one fragment, and valine, cysteine, tryptophan in another fragment. But since we have them in the same fragment, so the two cysteine must be linked by SS bonds, like this. The last fragment is isoleucine, phenylalanine, valine. When chymotrypsin cleaves after tryptophan and phenylalanine, this fragment is normally released. Now let's verify if our work is correct. In the given, we have that when the peptide is treated with 2-mercaptoethanol, it yields two polypeptide chains, X and Y. So, if this is the correct polypeptide chain, when we treat it with 2-mercaptoethanol that reduce the SS bonds, so it cleaves the bonds here and here, we will obtain two polypeptide chains. Now, when we treat the polypeptide chain with endopeptidase like chymotrypsin, the chymotrypsin that cleaves after phenylalanine, tyrosine, and tryptophan, cleaving the polypeptide after phenylalanine will release the fragment alanine, phenylalanine. And when it cleaves here, after phenylalanine, it will yield this fragment, glycine, cysteine, lysine. When it cleaves after the tryptophan, it will yield the fragment valine, isoleucine, phenylalanine. When it cleaves after tyrosine and of course after tryptophan, we will have this fragment here and the one here, both relating the cysteine. So this is how we solve such an exercise. Thank you for watching my video. If you like it, please like and subscribe.